Floor is all yours.
All right. Thank you, Brian. I'm really excited to give this talk to a bunch of awesome people studying kidney cancer, a fun and often neglected tumor type. Today, we're going to talk about my work that I did mostly as a PhD student, but with some translational aims as I returned to medical school. We're going to be talking about metabolism in the kidney cancer microenvironment, and it's not going to just be about VHL and HIF in the tumor cells. And as Brian mentioned, this is like my final Vanderbilt tour because as of Monday, I'm driving out to move back to Los Angeles and be a member of the UCLA medicine department as an intern. So that is my newest affiliation on this slide. As an MSTP student, I have a few less conflicts of interest than someone like Brian. So one time I got a flatbread from Agilent for discussing how to do in vivo ex vivo seahorse. And then I also got a Jimmy John sandwich from Miltenny for testing out some antibodies.
We need to work on this.
That's the next stage of growth in my career. And so what is important to acknowledge, which is why we're on this call with this type of program that Brian is growing, is that science is a team sport. Here's probably one of the best examples of a team. These are all Hall of Fame NBA players, I think, other than Christian Laettner. They settled aside their differences and just dominated. And no different is this project we're going to talk today, where Kim and Jeff Rathmel are two leaders in cancer biology, immune metabolism, RCC, and we put our heads together and developed a very cool and novel way to look at metabolism in mouse and human tumors. So today's talk, sorry about that, is we're going to have a brief background on immune metabolism in the tumor microenvironment. We'll go into two data sections. Data one has been published in 2021, but acts as a nice segue to some of the more translational aspects, which I'm sure there'll be some opinions and some input from this group. We'll summarize it and talk about what it means for the field of kidney cancer, probably IO biomarkers and future therapeutic development. So, 25 years ago, Bob Weinberg and Doug Hanahan developed the Hallmarks of Cancer. They updated in 2011. They've updated more recently. But what they called in 2011 were emerging hallmarks, were deregulating cellular energetics and avoiding immune destruction. This was before the clinical application, really, and success of CTLA-4, PD-1 blockade, as well as now in other tumor types, we have IDH1 inhibitors. But what we think about in the Rathmel lab, and what I'm biased to probably do future work, is that these two things are actually interconnected. Cancer cell energetics impact how immune cells function, but also cancer cells alter the immune energetics and thus their activation state. So there are many forces at play in this cycle, and we really like to examine these two things. And what becomes quite relevant is that the most common imaging modality in cancer, not kidney cancer, but across all cancer patients, is FDG PET. So this is a patient who has metastatic esophageal carcinoma, and we see that the primary tumor is located in the mediastinum, and there's tumors all in the liver, lymph nodes in the thorax area, and even he's having some iliac intensity. So how does this modality work? It's quite simple. It's a modified version of glucose that has this fluorinated F18 molecule. So you can use tracer amounts, little tiny amounts, and you see the activity of glycolysis. Molecularly, this is thought to be occurring because the FDG, just like glucose, goes through GLUT1. It gets phosphorylated by these hexokinase enzymes. Don't forget about this little detail. And then it marks which cells are undergoing glycolysis. Now, I think we're all pretty aware that cancer gene mutations promote glycolysis, loss of VHL, KRAS mutation, P53 loss or mutation. But what has become a whole paradigm, which Jeff started 25 years ago, is that immune cells, when active, also increase their glycolytic capacity and function. And so it's not a surprise that conditions like rheumatoid arthritis are glucose avid as well. So this is a patient pre-therapy, and they notice that there's localized intensities in their synovium, in the joints in the hand and the joints in the elbow, indicative that you don't need cancer cells present in the microenvironment to get FDG avidity. And this kind of provoked us to ask questions about, well, when a patient's esophageal carcinoma lights up, is it all tumor cells? Is it all cancer cells? Is it all fibroblasts? Is it all macrophages? And we started on that point because we were collecting what we colloquially reference as tumor juice or tumor interstitial fluid. And this was with the help of Matt Vanderheyden's group, the postdoc Alex Muir, who's now at the University of Chicago, and MD year-out student Ali Ahmed, who's now at UT Houston as an intern. And you spin the tumors over basically a piece of cheesecloth pretty slowly, collect the fluid, and send it for mass spec. And what we see both in human tumors and mouse tumors is that when you compare either the normal kidney parenchyma to the tumor or the plasma to the tumor, there's still abundant levels of glucose. This is glucose in the range of DMEM, RPMI, a different type of media, has about 20 to 25 millimolar glucose. So when labs have studied glucose restriction, they do it in the nanomolar or micromolar range. And we just don't see that when we study actual human tumors. And thus, we wanted to ask the question that if glucose is present in this heterogeneous tumor with a bunch of different cell types, who's actually consuming it? Do we see that cancer cells, as Warburg in the field kind of postulates, are the dominant per cell glucose consumer, or is there some immune cell or multiple immune cell populations that consume glucose as the field of immunometabolism has grown? And to do so, we had to collaborate with a bunch of different people at Vanderbilt. So good news, if you give a mouse a tumor, it ends up PET avid on the flank, just like that esophageal carcinoma example I gave earlier. And working with experts in PET imaging, Charles Manning and his postdoc, Allison Cohen, who are now at MD Anderson, and Melissa Wolf in our lab, we all started to put our heads together and see how could we actually use this feature to see who consumes glucose. So we combined the PET imaging. We would, prior to harvest, inject mice with FDG, wait about 40 minutes for the FDG to label the tumor, make single cell suspensions, and then use commercially available Miltenyi beads to pick our favorite cells of interest. And then we take them on this high-tech, high-resolution Geiger counter and be able to tell which fractions were consuming the glucose. And just to show you that this purification scheme works, in this experiment with MC38, which is a black 6 mouse colorectal tumor, the whole tumor has about 70% CD45 negative tumor cells and 30% immune cells. When we use the beads, the cancer cell fraction is about 90% CD45, and the negative and the CD45 positive immune fraction is almost as pure as you can get without going to a flow cytometer. And then when you track the radioactivity, here I'm going to show the Ranka model because this is a kidney cancer group. You see some interesting features. So the first is that the original Warburg and FDG based viewpoints are in fact true. So if you compare the per cell avidity of a spleen to the tumor takes up about 5 to 10 times as much glucose as the spleen. So that's a good positive control. Our theology that tumors are environments where glucose is consumed is in fact true. However, what became surprising is that when we split up the whole tumor into CD45 negative and CD45 positive, we see that actually the immune cells, here a very heterogeneous population, are actually the dominant per cell consumer of glucose. And you're going to say, Bradley, this is a BALB-C model. You injected this in the flank of the mouse. I'm not going to go through all the data, but we observe this when we inject the Ranka into the flank, into the kidney of the mouse. We observe this in MC38, the black 6 model, which I'm going to show more data from. We even did inflammatory models of carcinogenesis and see the same effect as also as genetically viral driven cancers. So we constantly see that there's immune cells consuming glucose in the tumor microenvironment.
Now I have a different question. the genetics of all the models are VHL proficient, even though you would assume physiological hypoxia to some extent promoting HIF activity. Have you compared just by IHC the extent of HIF activation in these tumors versus a VHL deficient tumor? Because our classical Warburgian effect is very HIF centric, right?
Yeah. So that is a story that's coming out of a graduate student in our lab, Melissa. I originally made the cell lines and gave them to her. And what appears slightly paradoxical is that if you make a VHL deficient cell line, you have no difference in CD45 metabolism, but negative metabolism. But you do have differences in myeloid metabolism, as if secondary changes, probably some soluble factors, like we do see elevations of myeloid chemotactic factors and myeloid differentiation factors, drive increased glucose metabolism. Now, that data is close to being out, and I wonder what people's perspective will be on that. But and that may be because when we knock out VHL, at least in these HIF1, HIF2 double positive cell lines, they don't seem to grow any faster. If anything, sometimes their proliferation is slowed. So it may take another graduate student or postdoc to start to unwind that. But VHL loss alone does increase whole tumor glucose uptake, but it actually seems to be myeloid specific, interestingly. But good question. So to dig a little deeper, because, you know, CD45 is a very vague term that marks all of the hematopoietic born cells, we decided to go on a little treasure hunt and find out who were the glucose consuming cells. These were all done on this slide in MC38. We replicated most of the findings in the Ranka model as well. And so what you see here is, you know, Jeff, being a T cell biologist, was like, pull out the T cells. I'm sure they're active. They're consuming glucose. And what we actually see when we purified the T cells first and then the remaining CD45 cells, it was actually the remaining CD45 cells that were glucose consuming. So that hypothesis didn't seem to be true. Then we went after a more targeted pull down of these other CD45 cells. We use CD11B, a pan-myeloid marker that goes up as cells really start to leave the bone marrow. And what we see is that myeloid cells that are CD11B positive in the tumor microenvironment are taking up huge, you know, we're talking about probably 5 to 8-fold more than the CD45 negative cells on a per cell basis. And then, you know, CD11B also is pretty vague, or it's not very specific. So we looked at both mature and immature cells in the MC38 tumor microenvironment, and they both seemed avid. So whether or not you're more MDSC-like with GR1 or you're more mature, we looked at F480 is higher in these cells, they're still consuming glucose. And we also specifically pulled down the macrophages, which in mice are F480 positive. This is not a marker in human disease. They, again, not surprisingly, are glucose consuming. Again, this could all be artifact of labeling techniques, of many things that happen on these days. So we wanted to use another technology to really confirm, or at least analogously support our results. So we did the seahorse on these same ex vivo purified populations. And what you see here in terms of their oxidative metabolism of the red F480 positive cells is much greater than the other cells. And I didn't include the ECAR tracings, but that appears to be the case well, that these myeloid cells are just souped up in comparison to all the other cells in the tumor microenvironment. And you may ask what drives this. So we did NanoString metabolism transcriptomics on purified populations. This time we used the FACS sorter just because we could get much more pure populations to get high quality RNA. And what we see when we do a PCA analysis is that when you're just looking at metabolism genes, we did not include CD4 or CD11. It was like a list of 400 of the 800 genes. The cells self-segregate. So you have a myeloid island up here in the reds and yellows. You have a lymphoid island in terms of CD4s and CD8s, and then the tumor cells are off in their own region. And then when you do a GSCA analysis of what are the pathways unique to those different groups, We color-coded them here, and to no surprise, both the immature MDSCs and mature myeloid cells are rich in these glucose and glycolysis-dependent pathways, whether they're hexosamine biosynthesis or carbohydrate synthesis. And actually, a group from China has shown that tumor-associated macrophages require glycation of immunosuppressive caspasins. And if they lose the ability to do glycolysis, they're no longer suppressive. So this original finding now, a year and a half, 2 years later, has been in a totally different system, has been shown that macrophage glycolysis or myeloid glycolysis is crucial to their suppressive function. There is much more in the paper. There's a whole story about glutamine metabolism. There's more transcriptomics, more flow cytometry, but I'm going to just stop it there just to kind of give a little summary. Is just like how we consume food, that where we grow up and who we're exposed to dictates what food we find interesting. I grew up in LA exposed to, tortillas and nopales, cactus. And so now I go to the grocery store here in Nashville buying cactus and people think I'm a little wonky. Cells in the tumor and microenvironment have similar metabolic preferences. So a myeloid cell born in the bone marrow with its unique program really likes glucose and needs it for its functioning. And a tumor cell, as the paper goes into, has a preference for glutamine. And so kind of a kitschy but easy to understand way is just to think about your nutrients that are on the side of your favorite cereal box. And basically each one of these groups has a preference for a certain cell type, and that's probably based on their cell of origin and what program they express with that. So, thoughts, questions before we move to the unpublished stuff. So, with that fact that
can I ask you a question about the control? Why did you choose spleen as your comparison to the tumor and?
Because we know the spleen at baseline is not avid, and we know that it's home of unactive T cells. There are not a lot of myeloid cells. We have done in the AOM DSS, we do have a non-affected like single cells from the colon, and they're also not avid. That's not tumor involved. But just many of these models, when we did the further purification, were all flank induced that or flank injected. That just is an easy control. The other problem sometimes becomes like if you did the kidney, to process the kidney in order to get diverse cells, it takes longer and longer and has this Percol gradient, which is really rich in glucose. And so we thought that that could screw up the results. So we just focused on getting the tumor cells themselves.
If you took a tissue that at baseline is FDG avid,
yeah,
would you see that the immune cells in that tissue are also resembling the tumor cell? In other words, are you simply seeing a tumor immune infiltration signature versus a metabolic activity signature?
No, so that's perfect segue into what I'm going to show. But a group probably a year after us was looking at Alzheimer's disease in mouse brains in humans. And they show that the FDG avidity in those systems and even in humans is correlated to microglial activation. So both at baseline and in Alzheimer's. So yeah, and microglia are CD11b positive. They're born in the bone marrow. So this is, I think, part of the myeloid life cycle, and we have some other projects in the Rathmel lab that seem to show that as myeloid cells differentiate, both in the normal situation and in inflammation, they see more and more glucose consuming to support that. Good question. So, we wanted to find potentially the mechanism of why there was this increased glycolysis in myeloid cells at a much more granular level. So we went back to the transcriptomics, which was a big help from fellow MD PhD Margaret Axelrod, who is world-renowned in the molecular mechanisms of IRAEs and is going to watch you for her pathology residency. And we just decided to compare the CD11B fractions So if we return to that PCA plot, basically, what are the top 20 genes in this group in comparison to everybody else? And you would not be surprised that almost all of these genes are genes that are related to myeloid cells. They're FC receptors, ApoE1, CD11B is ITGAM, CD14. But interestingly, there are a host of metabolism genes that are unique to myeloid cells. Two of them, are probably not that surprising. One is kynurenine metabolism, and that's been implicated in some of the IDO story. And then another one is thromboxane synthesis, and this underlies, the cardiovascular role of macrophages in myeloid cells. But there was one gene that's responsible for fixing glucose. It's a rate-limiting glycolytic enzyme, and it's hexokinase 3. So now if we look at the three main hexokinase isoforms, we see that hexokinase 1, known as a housekeeping gene, really doesn't have much cell type selectivity. I don't know why the significant stars disappeared. Hexokinase 2 appears higher in the myeloid cells, which tracks with data in our paper that phospho-S6 is higher in the myeloid cells, and hexokinase 2 is known as a HIF target, as people like Celeste have studied in kidney cancer for a long time. But hexokinase 3 shows this remarkable enrichment in myeloid cells, where it is virtually undetectable anywhere else. And even if you looked at the whole tumor as a control, this gene barely is on your radar. So what the heck is hexokinase 3? What has anyone ever done or thought about this gene? So I'm going to return to that figure that I talked about at the beginning of the talk, the hypothesized mechanism of FDG avidity. We're focusing on this rate-limiting first step in glycolysis, basically how cells get from doing facilitated diffusion to holding on to their own glucose. Here are the structures of hexokinase 1, 2, and 3. And what was previously appreciated is that hexokinase 1 is a great housekeeping gene. Many labs, people use this to normalize RNA sequencing data. As I discuss, hexokinase 2 is this mTOR stimulated gene. It's actually known to be like the most upregulated gene with T cell activation, because with ligation of the T cell receptor, up goes HIF and up goes their metabolism. And hexokinase 3, we really have little appreciation. You got to hunt through the literature to find something that talks about all these isoforms. And here's a review that compares the activity of these enzymes. And what you see is that hexokinase 3 actually does have slightly more affinity for glucose, making it a nice fit for the story we saw in terms of upregulated glucose metabolism. It does demonstrate this weird substrate inhibition. So it's not what you typically think of where the end product inhibits the enzyme at pretty high at concentrations that are in the tumor between 0.5 and 2 millimolar. Hexokinase 3 activity probably is decreasing, but it does have this affinity that in localized regions of glucose restriction or hypoxia, you would believe that cells that are hexokinase 3 positive would be scavenging the glucose. And interestingly, when you look at names of hexokinase 3, there is a vestige of maybe what phenotype we're seeing, where it was referred to as the white cell hexokinase or the CD45 positive hexokinase. But that may not describe the phenomenon we are in fact seeing. So first step first is let's validate our NanoString analysis. So we used here the bone marrow as a good control, and we got hexokinase 3 knockout mice and this first hexokinase 3 specific antibody. And what we see here is that sure enough, hexokinase 3 is expressed in the bone marrow, and in our knockout mice, it's undetectable. If you purify the CD11B positives from the tumor using our bead scheme, we have hexokinase 3 positivity. The knockout animals demonstrate, no signal. And interestingly, in the spleen and the tumor in wild type animals, you really don't detect this gene, very similar to the RNA analysis, that this gene is super enriched in the myeloid cells and is a part of their program, is a part of their being and livelihood. Now, all this data is in mouse tumors, which, really nicely pointed out by Abhishek, that, that's only one small part of human physiology. Let's see what happens across human biology. So in humans, just sitting there, all of us, we have lungs, and our lungs actually have a myeloid population at baseline, these alveolar macrophages. And sure enough, if you look at RNA sequencing of the lungs, CD11b perfectly correlates with hexokinase 3 expression. However, hexokinase 3 expression does not correlate to EBCAM, the dominant cell population in your lungs, to CD3, so any T cell population, or CD20. So the term of white cell hexokinase appears to be too broad. This is a proxy way to look at this at RNA. Most of us are now familiar with single cell RNA sequencing. So if you analyze just a data set that's put out into the world by 10x on PBMC, hexokinase 3, again, is only detectable in the myeloid cells and absolutely none of the other cells in your bone marrow. RNA does not always equal protein, even though that doesn't seem to, our MC38 example on the prior few slides seems to argue otherwise. And what we'll see here now is proteomics data. This is using mass spec, and we'll just use hexokinase 1 and hexokinase 2 as an example. I told you that hexokinase 1 is a housekeeping gene. It's pretty evenly expressed across all these different immune cell subsets. Hexokinase 2 is this TOR-related activation hexokinase. And sure enough, you see in CD4 naives as well as CD8 naives really low levels, and it increases with TCR stimulation. And then when you look at hexokinase 3 levels, the only cells that you detected in are CD11b positive, being eosinophils, but the dominant expressor is macrophages. So here we're seeing that hexokinase 3 seems to resemble a myeloid-specific gene across paradigms. Now, all of our tumor data so far has been in mouse. I'll just show you briefly that it is in fact true in human tumors, as well as human homeostasis. So this is data from Joanna Joyce's lab, where they did something very similar to us. They did flow cytometry. They got their populations of interest, which were microglia, CD45 negative tumor cells, T cells, macrophages, and you only see expression of HK3 in CD11b positive cells, whether they be resident population or whether they be monocyte-derived macrophages. And that's the same whether or not it's glioma or it's a brain metastasis. In this data set, there's a few kidney cancers, there's a few lung cancers, there's a few breast cancers and melanomas. And interestingly, this was done at both MSK and in Lucerne. So this is very strong, compelling data that hexokinase 3 is myeloid specific in tumors. Additionally, there's a database that Shirley Liu made before she left the Harvard system. I think she was at the Broad. And it allows you to query single cell RNA sequencing, which again, we get many cells, but we don't get much depth. And we see a very strikingly similar phenomenon. So I used these lighter colors to indicate lymphoid lineages, and you see no expression of hexokinase 3. These reds and browns represent myeloid populations, whether they be neutrophils or macrophages, and you see expression. There's only one type where you see tumor-specific hexokinase 3 expression, and that's actually acute myeloid leukemia. Those cells are CD11b positive. And then this breast cancer data, these myofibroblasts are also CD11b positive. So unless they're supposed to be myelofibroblasts, they're a misannotation and thus demonstrate that there is really a tight relationship between a myeloid program and induction of hexokinase 3. Now, what happens when we're inflamed or when there's pathology? You know, tumors are only one type, and we would like to maybe understand myeloid glycolysis across multiple paradigms. So we worked with Javed Moslehi's group while he was here at Vanderbilt, and we did our qPCR on heart tissue post myocardial infarction, and both in the acute phase, which is known to have an enrichment of neutrophils and macrophages, but in the chronic reparative phase, we still see elevation of hexokinase 3, and this does not occur with the other hexokinase isoforms, which remember carry out the same exact enzymatic reaction. And so I thank Javed and his postdoc Juan, who's now both at UCSF. And then we also see a similar phenomenon in ulcerative colitis. So this is a data set where they actually have over 10,000 IBD patients, and you can query your favorite gene. And in these three panels, these are all tissue-based RNA sequencing. So you're looking at the colons and the ileums and jejunums of patients who have inflammation, and hexokinase 3 is elevated. Now, that's probably not shocking now that I've showed you that it's myeloid specific, and key pathological features that make these diseases is that they have more blue cells, tiny blue cells in them. But what I did find striking is that there's really only 3 significant genes when they flow sorted myeloid cells from the blood, and the 4th or 5th most significant gene is hexokinase 3, and it's not significant because the FDR is set at 1 × 10⁻10th. So I started to wonder whether or not this is not just the presence of these cells, but it's them experiencing some sort of inflammation or signal. And what maybe confirmed that that was the case is all of this recent depositing of COVID data. And as I've previously mentioned, increased glucose metabolism is a sign of activation of immune cells. So in this study, they did RNA sequencing on whole blood from COVID patients. So GLUT1, which Jeff has studied for decades and demonstrates is a key driver of T cell pathology, T cell function, it actually demonstrates no correlation with any COVID status. So here on the left are healthy patients. And as we go to these 3 box whisker plots, this is basically increasing in COVID severity on the floor. Then these blue dots are COVID patients that are sick at home, which probably represents a majority of people now that we have vaccines and antivirals. And then this last column are patients who are in the ICU, and I think over 50 or 60% of them passed away. And GLUT1 as a gene regulated by HIF, induced inactive immune cells, does not show any relationship with COVID status. But strikingly, hexokinase 3 does. So as patients get sicker on the floor, hexokinase 3 goes up. And as patients enter the ICU, it reaches its maximal point. But patients who have COVID at home and have minimal, have significant symptoms but don't need higher levels of medical care actually are able to have this gene at a low level. So this, along with the ulcerative colitis blood data, beg the question, is there some sort of substance that can regulate hexokinase 3? It's not just a myeloid-specific gene. So the perks of being at a cancer center where there's a bunch of labs on your floor is you ask everybody for your favorite cytokine, give them to some macrophages, and see if they induce hexokinase 3. And voila, it becomes very striking what occurs is that hexokinase 3 appears to be regulated by interferon gamma. Other interferons that I gave to these cells do not regulate it. IL-1 beta, known to be a pathogenic myeloid factor, does not seem to activate it. But any combination of interferon gamma alone or with some of its friends seems to upregulate hexokinase 3. This is the RNA level. We will use that novel antibody that we got from Hussein's lab and show that absolutely strikingly it elevates in these bone marrow derived macrophages to a substantial level with providing interferon gamma. And so I thank Hussein and his former postdoc Farnaz, who shared their protocols and their tools with us. So this was at least strong evidence that interferon can induce hexokinase 3. But you may ask a question that in the tumor microenvironment, there's interferon gamma and there's cancer cells. So we got to test whether or not we can induce hexokinase 3 on those cancer cells. So on the far left, we have our immortalized macrophages. We have the MC38 colorectal model. We have B16 and LLC as other Black 6 models, all on a dish. And what we see quite nicely is that hexokinase 1 is a beautiful housekeeping gene. Its level is consistent among all 4 different cells. Like each cell line is a good control for itself. When we give interferon, we do see some induction of hexokinase 2. That probably returns to some of the work that was done by Leonis Platanius, who's now the head of the Cancer Center at Northwestern, where he shows that there is, in fact, crosstalk between mTOR signaling and interferon signaling, and maybe drives some of the reason why everolimus or mTOR inhibitors could work in multiple diseases because it's downstream of inflammatory cytokines. But what becomes remarkably stark is that hexokinase 3 is only induced in the myeloid cells when given interferon. So this seemed to be pretty striking evidence that it is a myeloid-specific interferon-stimulated gene. Now, some of you may ask, is the interferon working? So I'm going to show you a good positive control. I did flow cytometry on all of these cells, looking for PD-L1 expression. And sure enough, when you give interferon, the macrophages as well as all the cell lines increase PD-L1 expression. And the delta in PD-L1 expression is like very great in B16, much greater than the macrophages. So demonstrating that there are like different regulatory and transcriptomic circuits in cells of different origins. And just to close the loop, just to make sure that this is specific and not some kooky interaction between all these factors, we gave ruxolitinib and interferon. Ruxolitinib on its own does not change hexokinase 3. Hexokinase 3 in this plot is this middle band. Essentially, as we reuse the antibody, we get some more non-specific bands, but this is the right size and demonstrates the pattern that we saw previously. And so when you give uptitrating doses of ruxolitinib, you get decreased levels in hexokinase 3, where Rux is a clinically approved JAK1, JAK2 inhibitor. Now, that was all in some immortalized mouse models, whether they be cancer cells, whether they be macrophages. So I just want to show you a little bit of evidence that this probably happens in humans too. So this is from Jason Buenrostro's group, which is also at Harvard, where he does this amazing single cell attack-seq and other types of skills. And he usually shows his data in totally crazy formats. I'm going to show one that I think more people here are familiar with. So we're just going to look at tracks. And here is the hexokinase 3 gene, and the right side is the hexokinase 3 promoter. And what becomes very clear is that the promoter really only looks like an open promoter. This is using single cell attack-seq in the monocytes and the CDC1s. And basically in all these other cell types, there's not this appropriately open chromatin. But what's even cooler that Jason did in this experiment, as if he knew this project existed, is he gave these cells interferon LPS or a nonspecific stimulus like PMA. And what you see in the promoter of hexokinase 3 is quite striking and quite perfect, is that interferon specifically in comparison to LPS induces chromatin openness and then leads to expression of this gene. He calls these DORCs, domains of regulatory chromatin. It's a very cool technique, but just wanted to show you that, you know, our mouse data is correlated with some human features. So from that section, I think we have pretty convincing data that in mice and humans, there's unique transcriptional regulation that may make hexokinase 3 a more important gene than just a white blood cell hexokinase. It's only expressed in myeloid cells, and it's interferon gamma stimulated. So I haven't said anything about kidney cancer, and we're in a kidney cancer meeting, so I better fix that feature. So as you know, David has been a leader, Ari Hakimi and other groups are really diving deep into the myeloid cells. I hinted that our group has a impressive story on VHL, and Jeff is working on a story about obesity, kidney cancer, and macrophages. Why is everybody so excited about macrophages and kidney cancer? Well, if you look at CD68 expression, which is a key macrophage identity gene, it's highest in kidney cancer. The only disease where it's higher is diffuse large cell B cell, where they're doing RNA sequencing on lymph nodes, which have lots of macrophages at baseline. Agi Dekubas, who's a postdoc in our lab, now has his own lab at MUSC, helped me. And to validate this finding, Emily Arner, a new postdoc in our lab while I was in medical school, did CD68 on a TMA that we have in our lab, and we see that CD68 expression could be quite abundant and does seem to track with stage of disease. We have a limited amount of stage 4 patients, but this is in line with many published papers that other people have shown that more myeloid cells, more badness, and kidney cancer. So hexokinase 1 and 2 studied in kidney cancer all the time because we're talking about glucose metabolism. Hexokinase 3 often overlooked. So what happens to hexokinase 3 when you look at the protein level in kidney cancer? Super high. It's higher in stage 4 disease, so potentially discriminating between metastatic and non-metastatic disease. And interestingly, hexokinase 3 does demonstrate poor prognostic significance, and this is unique to this enzyme, where hexokinase 1 and 2 do not do that. Again, even though they catalyze the same metabolic reaction. So when you learn biochemistry, there is a lot more going on here than just glucose in the cell, and the cell makes ATP. The regulation in which cells express these enzymes really can have phenomenal impacts on our patient outcomes and maybe patient prognostication. So we wanted to just validate that the TCGA features were in line with things that we study in our lab. So we know that Chuck Drake's group and our group at Vanderbilt had discovered that these TREM2 high C1q macrophages are poor predictors of outcome where patients have advanced disease and perish early. So we wanted to look at hexokinase 3, comparing the TREM2 high to the TREM2 low patients, and in line with our hypothesis, you see more hexokinase 3 in the TREM2 high patients than the TREM2 low patients. Again, demonstrating that this is part of some bad, evil actor myeloid program. And I want to thank, the multiple collaborators to get, complicated science like this easy to access. So Scott Haig, Katie Beckerman, Brian, Alex Obravadic, and Chuck Drake all made this super easy and seamless to query. We then did a GSCA analysis asking, okay, what's unique to hexokinase 3 high tumors rather than hexokinase 1 and 2? And really what you see in hexokinase 1 and 2 high tumors is a gamush of metabolism and inflammation, which is maybe what you'd expect. But uniquely in hexokinase 3, there's only 3 pathways that are enriched, and they're all inflammation related. And thus, when you do cybersort on hexokinase 3 high tumors, you see cell subsets that have been shown to be bad in kidney cancer. There's more macrophages, there's more neutrophils, there's more CD8s, and there's more M2 macrophages. So this gene fits in line with kind of the current understanding of myeloid cells in kidney cancer, at least to me. If someone wants to tell me differently, happy to say more or less. And where we start to, where it bores out even more is in the neoadjuvant space. So Ben Vincent at UNC and Kim were working on this data probably 10 or 15 years ago with the help of Chris Wood at MD Anderson, where they were doing RNA-seq on tumors pre-treatment and post-treatment with the VEGF-TKI pazopanib. And what you see here again underscores that hexokinase 3 is really different than 1 and 2. So what we see here is that there's a treatment effect on a log scale of hexokinase 3. There's an increased expression of this enzymes post-VEGF treatment. I would think it was a few months. And that's not the case with hexokinase 1 or hexokinase 2. The scales on these are not all the same, which is a little unfair to hexokinase 3, but you see that there's a significant difference between the pre and post treatment, demonstrating that VEGF inhibition, which we know has some myeloid repolarizing features, is altering the hexokinase 3 expression. And in a newer age of genomics, we can use single cell genomics to have some sort of insight into what these hexokinase 3 positive cells are. It's not just one transcript in a large field. So using this paper from Ellie Van Alen's group and reanalyzing the data, we see that hexokinase 3, this is a whole TME picture. So down here are the epithelial cells, these are the myeloid cells, and I believe these down here are the lymphoid cells. And there's only hexokinase 3 transcript, again, myeloid cells. It's a subset of myeloid cells. It's not all the players. And what's interesting is that 2 of the top 4 genes are IFIDMs. They're interferon stimulated genes. So we really think that this paradigm that we tested in vitro is happening in our human RCC tumors. And this work was very helpfully done with the help of Logan Vlatch, who's a new graduate student in our lab, uncoding some of the biology around SETD2, which I think we all know is a long-held favorite of Kim Rathmos. So what the heck do we do with interferon response genes in the era of immunotherapy? I don't really know, but I'll just show a little bit of the data and maybe some of the controversy. So this is the current paradigm of cancer biology we're in. We give patients antibodies that induce the immune attack, and these activated T cells kill people's tumor cells. In melanoma, this works gangbusters, 50, 60%. We still got to find a way to treat those other patients. In kidney cancer, we have durable effects, but they're not as long lasting and curative. And this immune attack is induced by genes like interferon gamma and perforin. So interestingly, in 2019, Andy Min's group at Penn showed that actually it's not just interferon gamma itself. You need the gamma signaling in immune cells that predicts response. If gamma signals in your cancer cells, that's bad news because they upregulate PD-L1 and other factors. In 2020, Tony Rebus's group actually says it's not so complicated. If you have more interferons, great, these patients are responding, keep them on therapy. And again, this is all in melanoma. And now most recently, two students very nicely from Yale actually show that this story is way more complicated, that, we've developed all these CRISPR screens in vitro and in vivo, and the effects of interferon genes in vitro are quite divergent than they are in vivo. So I'm just going to say, if you find an interferon stimulated gene and you're interested in it, you got to do the studies. You got to look in large data sets to be convinced one way or the other. And it's probably tumor type dependent, but which allows us to say, okay, now we're going to investigate hexokinase 3 in kidney cancer. So to do so, I was lucky enough to work with Brian and Scott to be able to look at the Motzer subset data. So I'm sure everyone is very familiar with this. Patients in Emotion 151 were randomized to Bev plus atezo or sunitinib alone. And we see that there's probably 2 or 3 interesting clusters from a predicting IO perspective. That clusters 4 and cluster 6 really have these remarkable benefits of these drugs. And those are patients that we got to really try and find early. And Brian and Scott are running optic to try and do some of this. And when you look at the paper, there's interesting features of cluster 4, where cluster 4 has both myeloid infiltrate and interferon gamma. And cluster 6 has just myeloid genes, but really no interferon signaling. So you can generate a hypothesis that maybe we'd see hexokinase 3-1 generally higher in 4 and 6, and maybe we'd even see it higher in 4 than 6 because it has the interferon, the driver of that increased hexokinase 3. So we just first did some clustering of the hexokinases and myeloid genes. And what you see really nicely is that hexokinase 1 and 2 are totally independent of all these myeloid factors that multiple groups have shown are bad in kidney cancer. Whereas hexokinase 3 is smack dab in the middle with CD68, TREM2, the C1q isoforms, CD11b, thromboxane synthesis, which our other analysis showed is myeloid specific. So we're really seeing quite profoundly that in human kidney cancers, hexokinase 3 is a part of this myeloid program. And then when we look individually across the subsets, hexokinase 1 and 2 don't really show, and these are like log 2 normalized variance counts, whereas hexokinase 3 does demonstrate an enrichment in those immune responsive groups. We're currently working on doing survival analysis to show that they benefit in a treatment-related form or not, but I think this is intriguing in a future for this gene or genes like it. And
hey, Brent, let me interrupt. Can I interrupt
Yeah.
for 1 second? Sorry. So,
Yeah.
In our optic trial, we're treating 4 and 5 with ipi-nivo, right?
Yeah.
6 we're excluding.
Yeah.
Because at least in the Emotion 151, it wasn't, 3 and 6 were the least responsive to anything and probably driven by other things. So I'm just trying to jive that with what you just presented.
Well, yeah. So I think what is unique to 6 is that they do have myeloid cells, but they're very different myeloid cells. Okay. I guess would be. And they're predominantly what you see here, and I've talked with Scott about, they're predominantly the complement expressing ones. So more in line with the things that were in the Truck-Drake paper and Califo and things to think about. So that's why when Scott, when the arms of it are mobile, or, when Scott was talking to the TREM2 people, maybe that is upstream of those factors. That may be an agent where those patients benefit. And I think that's a bigger question for myeloid targeting in tumors, is that it's not going to be a one size fit all, because the myeloid cells exist on such a spectrum. And I know that Jed Walchuk's group is trying to publish their PI3 kinase gamma story. And they see patients do respond in the refractory PD-1 melanoma world, but it's not everybody, and it's not everybody with myeloid cells. So there needs to be some distillation of those features in each group to find better targets.
Fair enough. I'm not sure if you're almost done. I just want to have a few minutes for questions too.
Yeah. This is my last data slide. And then just to say, in line with the Chow data, this is way more complicated. So you'd say, oh, great, hexokinase 3 immune stimulated. That doesn't seem to be the case in melanoma, where it's not really telling us anything about patient CRs or PRs. This is a Vanderbilt cohort, but you can also go online and use this cancer immune database, and we see really no effect of hexokinase 3 in responders or non-responders. So we think that this is an RCC specific effect, and that it's acting potentially as an AND gate, in that it senses inflammation and it demonstrates that there's some appropriate myeloid population in the tumor microenvironment. And this, I think, is reminiscent. I'm sorry if this comes out really poorly. Vanderbilt kicked me off its journal access today, so I couldn't get the nice figure. But to the McDermott story about T effector high and myeloid high, where the patients who benefited from atezo-bev and genes like hexokinase 3 may underlie that, because the T effector is driven by interferon. So future work is that survival analysis and then publication before Kim Rathmel maybe chops my head off, even though she's the nicest, kindest person in the world. And the implications, as Brian kind of hinted, is that, you know, there's a new age for metabolism inhibitors. It's not that they're going to kill every cancer cell, but maybe they can reprogram or reprime myeloid cells or other immune cell subsets. And I think another lesson from this type of work is isoform specific inhibitors, whether using degrons, better chemistry, or potentially siRNA. What you see from our data is that the T cells are actually really enriched for hexokinase 1 in a way that other cells aren't. So if you could develop strategies to specifically target hexokinase 2 and 3, you may be able to stop tumor growth and myeloid activity while allowing the T cells to function. So with that, I'm happy to take any questions. And if anyone wants collaboration or has more data, has more thoughts, you should definitely stop by at ASCO. I'm a member of the Checkpoints, and Tom Gajewski would kill me if I didn't advertise the band. So there's a show on the Sunday night from 8 to 12. We'll play a lot of dad rock cover band music and have a good time. So here's my contact.
Great. I appreciate it. There's just a couple minutes for questions from the group. Stunned silence has fallen over the crowd.
Okay, I'm going to ask. I wanted everybody to speak up before I did. I'm thinking mechanistically about what HK3 is doing. If it's a kinase dead version of just HK1 and HK2, that's not sufficient to explain the biological phenotypes that you're seeing. What in your mind is going on in terms of the functional role of this protein?
Yeah, so that's probably the most insane part of it. So the knockout mice have no phenotype other than no neutrophils. The tumors grow to the same size. We do the uptake assay, and they demonstrate no difference, which could be that there's just no neutrophils to study, and that's screwing that result. I've overexpressed the gene in both tumor cells and myeloid cells, and it actually causes the cells to die. So it really is complicated and would probably be a lab's worth of work. Now, somebody else's group made like a bio-ID hexokinase 3 and claim it binds the mitochondria, but that's actually the domain that the enzyme is missing. So I don't believe that that paper makes a whole lot of sense to me. And hexokinase 1 and 2 are known to regulate ROS via their mitochondrial binding domain and how they shuttle and use NAD and ATP. So I guess you could imagine that when myeloid cells experience interferon, they're doing a lot of phagocytosis and a lot of antigen presentation. And I think this has to be part of that program in terms of ROS protection, and maybe some of the TISH data supports high levels of neutrophils. Those cells actually have very few to no. As a neutrophil leaves the bone marrow, it loses its mitochondria, and thus we maybe think that in those cells it's playing a metabolic role, but those cells in our assay don't live long enough out of the animal to be properly accounted for. So I really think it's one of those projects where the unknown unknowns continue to grow, because the previous paradigms that, Vander Hayden or Celeste or Craig Thompson would put forth, that more glucose cells should proliferate more, is not the case with hexokinase 3.
Hey, bro, this is Ritesh. Really, really great talk. Just trying to think through like some of the work that you had mentioned about like ruxolitinib, particularly with hexokinase 3. I mean, ruxolitinib was also, it's interesting to kind of see some of the data with COVID, because if I remember correctly, there was published data, if I remember, in the Lancet about, small trials of ruxolitinib, particularly for severe COVID patients, without significant response. But do you see sort of a role for ruxolitinib, in thinking through myeloid targeting, or, and kind of the relationship with that, with immune checkpoint and maybe even IRAEs?
Yeah, I think there's a role that probably people previously overlooked for like subset specific targeting of small molecules, right? Like, wouldn't it be great if we could give people dual ICI and they could get VEGF inhibition, but it only happened in their neovasculature? And I think a similar thought should probably happen with JAK1 and JAK2. And Justin's group next to us on the hall kind of has similar vibes, where like, at first, if you give a JAK inhibitor with PD-1, you'd imagine like, oh, that's not going to work. But it's actually more complicated, because T cell interferon signaling is good for their activity, but to a point. So you really need to hit that gain knob on the head to get and you need to know what cells you need to inhibit the JAK stat in. Now, I am probably more of a believer or a fan of the Andy Min model, where you really need more immune related JAK1, JAK2 activation and less cancer cell. So if you could have some way where you could have tumor cell specific targeting of a small molecule, I think that would be a way forward. And interestingly, the Rux COVID story, that was negative. But baricitinib, which I believe is a more specific, either it hits one or two more than one, actually did show positivity, and we sometimes use it in our ICU here at Vanderbilt. So again, it's complicated. And Michael Savona's group here in AML really think they have drugs that as you increase their dose, they hit more JAK1 or JAK2, which would kind of be an interesting phenomenon, and maybe counteract some of our clinical phase 1 development where it's all maximal tolerable dose rather than some sort of biological endpoint.
Yeah, it's like, Rux it feels like it's kind of coming back a little bit, just because now I think there's a lot of interest in how it might play, JAK-STAT signaling might play a role in lineage plasticity in prostate cancers. And there's a, science paper that came out of Sawyer's lab here. Have you looked at like HK3 levels in sarcomatoid RCC?
I have not compared the subtypes, but we can do that easily from the TCGA, and I would imagine it's high.
It might be interesting, yeah, particularly with like the immune checkpoint story and things like that. But I don't know. I'm sure everyone here on the call has been sort of thinking through this, but like plasticity and sarcomatoid variants and rhabdoid variants, and there's nice work from David and the Dana-Farber group about some of that stuff. So if it's sort of it's like a nice corollary to just sort of think through.
Yeah. We definitely see in some of that data and some of the emotion data, the EMT genes. The EMT related factors and signatures are higher in 4 and 6. And what does that mean for either targeting?
Yeah, plasticity.
20 years ago when people were interested in these pathways, we never thought we could target a transcription factor or and I think that may play a role as well. And
hey, Brad, this is great. I'm going to have to close up to get to another call, but really appreciate your time and expertise, and look forward to continuing to collaborate even though you're leaving us.
Thank you for this time. And
Awesome.
If anybody has more questions or thoughts, please send them my way.
Yeah, good
and
luck
we'll
with the
go from
move.
there.
All right,
Good
thanks
talk.
everyone.
Thanks.
Thank you.